In this segment on the special senses, let us first begin with the olfactory pathway. Olfactory pathway deals with the special sense of smell. This information is carried by the first cranial nerve or the olfactory nerve. This olfactory pathway has some special characteristic features. Firstly, the cell bodies of the primary afferents, or what we call as first order neurons, they occupy the surface epithelium. In every other sensory pathway, the first order neurons were located within the ganglia, whereas here they form part of the surface epithelium, which will be lining the roof of the nose. Secondly, these primary afferent neurons undergo continuous turnover. That is, every 1 to 2 months, these cells will be shed off and new cells will replace the old ones. So unlike every other place where the neurons are present for the entire lifetime, here these cells are continuously undergoing turnover. Thirdly, the pathway projection to cortex in case of olfactory pathway is entirely ipsilateral. Every other ascending pathway either projects to the contralateral side, or in case of auditory pathway, it projects bilaterally, whereas olfactory pathway projects to the ipsilateral cortex. So what you smell through the right half of your nose will go to the right cerebral hemisphere. And finally, this is the only pathway that relays in the cortex without passing through thalamus. If you notice, every other ascending pathway first relays in thalamus, and then from thalamus it is projected to cerebral cortex. Whereas in case of olfactory pathway, it directly projects to cortex without going through thalamus before. First, let us consider initial part of the pathway, that is, it includes olfactory epithelium, olfactory nerves, olfactory bulb, and olfactory tract. Let us first concentrate on the figure shown on the left half of the screen. The olfactory epithelium lines the roof of the nasal cavity and also uppermost parts of the septum as well as lateral wall of the nose. Now this contains the primary afferent neurons, axons of which will join together to form 15 to 20 nerve rootlets. These are the olfactory nerve rootlets. It never forms a single nerve. Instead, it is broken down into 15 to 20 nerve rootlets. These rootlets will pass through the foramina in the cribriform plate of ethmoid bone to enter the cranial cavity in the anterior cranial fossa. Once they enter the cranial cavity, they relay in olfactory bulb. Now, this olfactory bulb is found on the undersurface of the frontal lobe on its orbital surface. Now, the axons which start from the olfactory bulb will then continue posteriorly as olfactory tract. So, let us consider more details about this in the picture shown on the right side. So, first we are showing the olfactory epithelium. It contains 3 types of cells. The first type of cell is the olfactory receptor neurons. These are bipolar neurons having dendrites facing the nasal cavity and axons traveling upwards towards the cranial cavity. Each dendrite, it has a single dendrite and single axon, that's why bipolar. So each dendrite will end up in a bulbous expansion, and that bulbous expansion shows multiple cilia. These cilia will contain surface receptors which will combine with odor molecules, and this chemical energy is then converted into electrical impulse, which is transmitted through these olfactory receptor neurons. The transmission, however, depends upon strength of the impulse. If it is strong enough to set an action potential, then it will be transmitted. As I said earlier, these olfactory receptor neurons have a lifespan of about 30 to 60 days only. So every 1 to 2 months, these old cells will be shed off and the new cells will replace their place. So the new cells are formed by the second type of cells which are located within the olfactory epithelium. They are called as basal stem cells. Being the stem cells, these will continuously be multiplying to replace the old and shed off olfactory receptor neurons. The third type of cells are the sustentacular cells. These will support both olfactory receptor neurons as well as the stem cells. So here we are seeing the cilia which will be having the surface receptor molecules which will combine with odor molecules. If the impulse is strong enough, then the impulse will be transmitted through the dendrite to the body to the axon, and these axons will join together to form 15 to 20 olfactory nerve rootlets on each side. There are a total of about 3 million axons on either side. These axons will combine together to form 15 to 20 olfactory nerve rootlets. So they pass through the cribriform plate, enter the cranial cavity, and then reach the olfactory bulb. Now within the olfactory bulb, these are 3 million axons. We have a type of cells there called as mitral cells. They are only about 50,000 cells. So there is convergence here. These axons will converge together within the olfactory bulb at the glomeruli, where they synapse with dendrites of these mitral cells. Now axons of these mitral cells will project backwards to form the olfactory tract. Apart from mitral cells, there are some other cells within the olfactory bulb. They include the tufted cells, granular cells, and inhibitory dopaminergic cells. And within the olfactory tract, apart from axons of the mitral cells, there are some other fibers also. One is the axons of the tufted cells are also found in the olfactory tract. And secondly, corticofugal fibers. These are the fibers starting from the cortex and reaching the olfactory bulb. There are also fibers coming from the opposite side olfactory tract here. So now the impulse it has started from the nose. So you get a smell. Now that smell is carried through multiple odor particles or odor molecules. They come and combine with the receptor molecules present on the cilia. If this information or impulse is strong enough, that will set in an action potential, which is carried by the olfactory receptor neurons to the glomeruli present in the olfactory bulb. Now, where these axons will synapse with the dendrites of mitral cells. Now, the mitral cells are carrying this information to the cerebral cortex via the olfactory tract. Let us see the rest of the pathway in this picture here. So here we are seeing the olfactory bulb continuing posteriorly as olfactory tract. If you trace this olfactory tract more posteriorly, just in front of the anterior perforated substance, it flattens to form olfactory trigone. Now at this olfactory trigone, this olfactory tract divides into 3 striae. There is a lateral stria which has got few neurons arranged along with it. Those neurons together is called as lateral olfactory gyrus. This lateral olfactory stria will relay in a gyrus known as gyrus semilunaris. This is underlying cortical part of the amygdaloid nucleus. Then we have intermediate stria, which may be absent in some people, that will end in olfactory tubercle, which is again located within the anterior perforated substance. And then there is medial olfactory stria. Again, there are neurons which are located along with this medial olfactory stria known as medial olfactory gyrus. Now this medial olfactory stria and the diagonal band of Broca, which is shown in blue color here, these two will travel on the medial surface of the cerebral hemispheres. They will relay either in the subcallosal area, or the fibers which are there in the medial olfactory stria will go to the contralateral bulb passing through the anterior commissure. Now this is important in suppressing information coming from the opposite half of the nose. Then we have what is known as band of Giacometti. Now we have said that the medial part of the parahippocampal gyrus anteriorly is separated by the renal sulcus into a small area known as uncus. Now within this uncus, there is this band of Giacometti, which is nothing but the tail of dentate gyrus, which we will be dealing with the limbic lobe video. Now that will separate this uncus into 2 parts, a posterior part known as intralimbic gyrus and an anterior part known as uncinate gyrus. More laterally, we have a large area known as entorhinal area or area number 28. This is the anterior part of the parahippocampal gyrus. And just lateral to the anterior perforated substance, we have gyrus ambiens. Now this is nothing but the area located at the Lyman insulae part of the insula, that is apex of the insula, So these are the areas which are found on the undersurface along the medial part of the temporal pole. So we have two terms here. One is prepiriform cortex. The prepiriform cortex includes lateral olfactory gyrus. What is lateral olfactory gyrus? These are the neurons which are located along the lateral olfactory stria, plus the gyrus ambiens. Gyrus ambiens is, as I said, it is the Lyman insulae area, which is basically located at the apex of the insula. So these two structures together, we call it as prepiriform cortex. Now, prepiriform cortex with periamygdaloid region. Periamygdaloid region includes gyrus semilunaris and all the three subparts of the uncus, plus the perirhinal area. And the laterally located entorhinal area, or the area number 28, these three parts together is known as piriform cortex. Now, this piriform cortex is the primary olfactory area. So all these areas which are shown in color on the left side, marking the anterior end of the parahippocampal gyrus, they will form what is known as piriform cortex, or the primary olfactory area. So the projection of the olfactory sensation to the piriform cortex is for conscious perception of smell. So once the information reaches this part of the cortex, as you notice, this information reached directly from the tract to the cortex without passing through the thalamus. Now, once it reaches this part of the cortex, the sense of smell is perceived. Then it projects this information to the orbitofrontal cortex, especially the posterior part of the orbitofrontal cortex. That is where odor discrimination happens. So it is the smell of a particular brand of perfume or a particular brand of soap, or a smell of coffee. So the the odor discrimination happens in the posterior part of orbitofrontal cortex. Information of smell is also shifted to hippocampus, which is located just behind. Now that is for encoding olfactory memory. In fact, olfactory sensation has a very strong memory encoding. What you have smelt even as a child will be remembered for the rest of your lifetime. So that is done by its projection to hippocampus. The information also reaches amygdala. That is for the emotions associated with the smell. So it's for the emotional responses. And then there is projection to even hypothalamus and brainstem reticular formation. That is via the medial forebrain bundle. Now that is for the visceral responses. With smell, it is associated with either salivation, gastric secretion. Sometimes a foul smell is associated with nausea, or it is associated with arousal, like early morning a whiff of coffee smell always wakes you up. So that is mediated by the projection of this sense of smell to hypothalamus and brainstem reticular formation. So what happens if there is lesion to any of these olfactory areas? Bilateral injury to all the olfactory nerve rootlets, that will result in anosmia or absence of smell. You will find that there is even absence of smell if there is common cold, because that coats that roof area, surface epithelium, with a coat of mucus, which will not allow the odor molecules to join with the receptor molecules on the cilia. Then there is a phenomenon known as parosmia or troposmia. That's distortion of quality of odor. So you are smelling something, but you'll smell it as something else. Now that is the parosmia. That's again because of the altered pathway through the tract. Then there is phantosmia, that is more of involvement of the temporal lobe. Here, temporal pole. Here actually there is perception of smell that does not exist. So a person is having olfactory hallucinations, or it could be even olfactory aura just preceding the seizures, temporal lobe epilepsy. That's known as phantosmia. So you're smelling something which is actually not there. And then there is, in case of olfactory lesion, you want to test for olfactory function. So for each half of the nostril, you will be asking the patient to smell some of the familiar smells, like smell of coffee, menthol, peppermint, and ask him to identify. So test it on one side first and test it on the other side to find out if there is any problem in either of the sides. But please do note, the sense of smell or the sensitivity to smell will reduce with aging. So thank you very much. Hope you enjoyed this video. Olfactory pathway is a simple yet very basic pathway, because especially in case of lower animals, that is the pathway which keeps the animals alive. If a deer can smell the tiger before the tiger smells deer, deer escapes. If tiger smells the deer before it is smelled by the deer, then tiger gets its food. So smell is a very strong special sense in lower animals. In human beings, however, it's one of the 5 special senses we have. Thank you very much. Hope you enjoyed this video. Please visit this site for other neuroanatomy videos. Thank you.